Good afternoon there, everybody. It's nice to be part of a wonderful, enthusiastic group. And as I always say, immunology is a fun field. Allergy is a very practical field. So when you take practical and fun and put together, then it becomes amazing. So hopefully what we'll do is appreciate how basic immunology affects the practice of allergy. And most important of all is it helps to understand disease mechanisms and some of the rationales of treatment that we use. And at the outset, I want to welcome all the candidates and say if you don't feel shy, if you have any questions, please do ask. And if I can't pick up your hand in the crowd, just stand up and then Dr. Gupta will coordinate it. So with that, let me get started. And what I would like to do this morning, from here, afternoon there, is to sort of walk you through some basic immunology, how it can be applied to the understanding of allergic diseases. So like everything in the world, you need a structure and a function. If you have an organization which is well structured and where all the members are interacting with one another, the output is excellent and it becomes a Fortune 500 company. Similarly, the immunology system, I would say, is a Fortune 1000 company because it does exceedingly well considering the complexity and how it can be challenged, both from changes within our own homeostatic pathways as well as the external world. So the immune system, I hope you can see the pointer, starts with a single cell that's called a pluripotent stem cell. I look at a pluripotent stem cell like a seed. And if you want a seed to grow well in a soil, you need the seed and a nutritious soil. Similarly, when the yolk sac in the child or the bone marrow cells develop, the cells are the pluripotent stem cells, depending on the kind of manure and the watering regimen you have, which in this case we'll call it as the cytokines and the stroma. So the stroma and the cytokines provide the nutrients and the water, and the seed is the pluripotent stem cell, and it can grow in many ways. For example, it can become the forerunner of the myeloid precursors from which you get your hematological cells, the red cells, the platelets, the basophils, and the eosinophils, and the granulocytes and monocytes. Or the pluripotent stem cell, when it is a different kind of microenvironment, which is chiefly driven by interleukin-7, becomes the precursor or the lymphoid progenitor cell. This cell will then go through various organs, like the thymus to become the thymic-derived T cells, which you will have heard about, as well as through the bone marrow, where it will become the B cell lineage. So we're going to be not only talking about the structure of the immune system, but how they function and how these various cells together can function as a cohesive fundamental unit. And derangements in this function often will give rise to a variety of diseases. So as we get started, one of the most important essential facts in immunology is to recognize that the immune system is always active, whether you're walking, sleeping, listening, and the elements of the immune system, both T cells, B cells, and the monocyte hematopoietic system are constantly interacting with the antigens which we release from the body. For example, every day, our red cells, white cells, platelets, epithelial cells, they die. They die by what's called a programmed cell death. And during that, they release the nucleic acids, they release the transcriptome proteins, and so these are called self-antigens. In addition, we also have circulating thyroid hormone, adrenal hormones. All these are called self-antigens, and the body does not respond to self. It will interact with them and sense them. At the same time, it also will recognize foreign alien antigens, such as bacteria, virus, fungi, and tumor cells, and it should interact with them and engage with them. So the immune system is a double-edged this.
Dr. Murali? Dr. Murali, can you hear me? Dr. Murali, can you hear me?
Self means antigens which we release every day in our body. For example, our red cells, platelets, epithelial cells, they die by aging, and during that we release DNA, RNA, histones, and those are called self antigens, besides the hormones like thyroid hormone or adrenal hormones or insulin. Non-self are bacteria, virus, fungi, and tumor cells which try to threaten us. So the important aspect of the immune system is it should interact with non-self antigens. You may want to call them pathogens or tumorogenic substances and eliminate them. If we don't, we end up getting immunodeficiency disease or tumors. On the other hand, if we have excessive response to self antigens such as our hormones or our DNA, you end up getting autoimmune disease, which could be thyroiditis, insulin antibody mediated diabetes, or lupus, etc. So, with this fine regulation to detect and respond to self and non self antigens is done by a very well regulated immune system, which is basically the basis of immune regulation. It is a communication between these various cells that is vital. And there are two ways we can communicate. For example, if was there in Delhi, I can shake hands with Dr. Niraj Gupta and have a direct hand-to-hand contact that will be called cognate interaction. Since was not fortunate to be there, I can at least communicate with you all via the internet, which is like cells and cytokines. So there are two ways. We can do by long distance without physical presence or by distant pathways. So both of these are utilized by the immune system. You will realize that the cognate interaction is vital when you process an antigen and get the initiation of the immune response. Later on, we can amplify by cells and cytokines, which can be at distant sites. The cell could be in the spleen, the immune response could be systemic. And we'll talk a little more about the network pathways as well as the cognate interaction as we go along. With this as a background, so one will say, what is the holy grail in allergy and immunology? The holy grail, or the ideal achievement which we aim for in a good immune system, is two ways. One is we have to induce immunological unresponsiveness. That means we see an antigen like double-stranded DNA or our own thyroid hormones and we don't respond to it. This is called tolerance. Tolerance means immune unresponsiveness brought out by an active interacting immune system. And for example, allergic diseases is a classical example. If you inhale Parthenium grass or in a person who has shrimp or prawn allergy, these allergens, we should tolerate it. If we don't tolerate it, you end up getting disease manifestation. So what we try to do as a therapy is to induce immunological unresponsiveness, which is one way by which allergy in immunotherapy works. You redesign the immune system or re-educate it so it will look at the antigen but not make the detrimental immune response. So immune unresponsiveness is a desired treatment holy grail for allergy. It is very important in the treatment of autoimmune disease. Here we have not achieved antigen-specific tolerance as yet, but we use medications, which could be immunosuppressive agents or biologicals, to modify the immune response. I think where it has been well exploited successfully is how transplantation immunology has become useful. Nowadays, it's pretty common to do bone marrow transplants, renal transplants without major adverse effects. So these are called allogenic tissues. Same species, human, but dissimilar genetic background. So we can give a kidney from an individual A to an individual B, as long as they are matched as best as we can. But little rejections can be modified by medications or biological agents. So there also you induce tolerance to the transplanted kidney. So you see the application of immunology in allergic diseases, in rheumatological diseases, in transplantation immunology. But what is making headlines these days is in oncology. There are more immunologists in a division of oncology than in an allergy immunology division, because tumors are common. And we do recognize that when the tumor grows, the tumor sheds antigens which paralyze the host immune system, both the T and the B cells. because it shuts off the immune surveillance, the tumor can continue to grow. What biologists and immunologists have understood is there are signals between the tumor cells and the human host T and B cells, which can be overcome by medications. So these signals are called inhibitory cognate signals. And the approach we use is they're also called checkpoint. You use drugs to inhibit these checkpoints, so you overcome the tumor inhibition of the host, so you unleash the host immune response. So the host T cells will now recover, go back and kill the tumor-specific antigens. So what you have is two important things. One is you need to turn off the immune system in allergic disease, autoimmune disease, and transplant. But in tumor immunology, you not only use chemotherapy to kill the cells, the residual tumor cells, which are turning off the immune system, can be made to be shut down by medications called checkpoint inhibitors. And I'll touch on those cognate signals as we go along. So this is where in the last 3 to 4 years, the whole field of tumor oncology has become tumor immunology. So how do cells and cytokines work? The long distance communication. Very easy. Whenever there's a stimulus, the stimulus binds to a receptor. This is like your antenna receiving signals, and the antenna communicates to your television set, and then you get a response. That television response is appreciated by other people in the audience. So there can be one or many cells which recognize it, and they react to this, and then you get biological effects. Some people may enjoy a movie, some people may think it's useless, some people may say, well, it teaches a good lesson. So the biological effects depend on what the end outcome is. So this is mediated by what's called cytokines. Cytokines to the immunologist is what hormones are to the endocrinologist. Every endocrinologist will not talk about thyroid without TSH, T3, T4, and the feedback. Similarly, immunologists will talk about cytokines and cytokine pathways. And these cytokines can act on the same cell. That is like your own self-education. You read a book or a chapter, you understand it, and you assimilate it, you analyze it. That's called autocrine. Or two friends and colleagues may sit together and discuss, and there are two dissimilar people, but they respond to the same signal. That's called paracrine. Or it could be distant. For example, like endocrine. And ACTH is released from the pituitary, walks through the adrenals and stimulates the adrenals, which is a few feet away. Or what we are doing now. I'm talking here through the beauty of the internet pathways. It reaches you so we can communicate. So you will very soon see that cytokines are in the afferent part of the immune response. So when you get a biological signal, you release cytokines, they talk to a cell, that cell makes cytokines, which then talks to an end organ like the lung or the kidney, and you bring about a desired or a detrimental immune response. So cytokines are exceedingly important network communicating molecules. So I'll summarize a little bit of basic immunology, which you probably know very well, but sometimes repetition awakens memory. The immune system is a great communication network. It can talk to one another by direct contact or via soluble factors. These soluble factors are called cytokines, and the cytokines bind to specific receptors. And this knowledge is very important. For example, there is a drug called dupilumab, which, as you all know, has made headlines in the treatment initially with atopic dermatitis. Now it's receiving approval for asthma. There are studies coming out on food allergy. And what this drug does is IL-4 and IL-13 can bind to mast cells, basophils, eosinophils, and activate them. But they have the same common alpha chain. So this drug blocks the IL-4 receptor alpha and IL-13 receptor alpha. So two signals which are driving these cells to cause allergic inflammation can be blocked. So you can appreciate how the biology of cytokine receptors has ended up in a very useful drug. And there are very many examples I can give, but I just thought we'll talk about this drug, which has really become one of the major therapeutic benefits in treating complicated and difficult allergic disease. However, its cost is prohibitive. That's another story. The other important concepts I want you is cytokines are pleiotropic, that is, they are multitasking. And the best example of this is probably PK, because he can talk about asthma, he can talk about food allergy, he can tell you stories, he can give you advice on life, and he is an organizer. So he can multitask. So he is a very pleiotropic cytokine. And then redundancy, that means sometimes we have too many bosses. But sometimes it's good because one boss misses something, the other boss takes over. For example, Dr. Gupta is organizing it beautifully. PK and Vinay have throwing in their expertise. So the three of them together make sure that the conference and the educational activities are done well. So you may say you don't need three people, one is enough. The answer is maybe, but it's good. Share and get the job done. And that's another thing the immune system does. If IL-4 fails, IL-13 will kick in. So that's why you have to block both receptors. The other important concept is there's synergism, which means cytokine A will combine with cytokine B and bring about inflammation, like IL-1, IL-6, TNF-alpha. On the other hand, gamma interferon will neutralize interleukin-4. Interleukin-4 causes allergy, gamma interferon turns it off, and vice versa. So there's both synergistic and antagonistic functions. So all these you're going to see coming into play. But how does this happen? It happens in the structure of an immune system, which is made up of two components. We are all blessed and born with what's called innate or inborn immune system. It comes with the conception of life. And these are cells and tissues which are present at birth and they function at birth, like the epithelial cells of the skin, the GI tract, the phagocytic cells, the white cells, the monocytes. And there are important cells which will probably be very vital as we move in the next year or two. These are called innate lymphoid cells. And there are three types. They are called ILC1, 2, and 3. ILC1 has innate ability to detect viruses and bacteria and respond, even though they don't have all the T cell receptor complications or complexities. ILC2 is involved in allergic disease in the GI tract. They are probably vital, which is why I can drink milk and not react. Somebody has a problem with milk allergy, drinks milk, and these cells respond in a detrimental fashion. Then ILC3s are regulatory cells, cells which turn off the pathway. So you can see a child is born with limited weaponry or limited ammunition, but can survive. Later on, when the child gets educated by either infections or coming in contact with siblings or the environment or the microbiota or by vaccination, you adapt or you educate the immune system, and that's the adaptive immune system made up of T cells and B cells. So we will touch a little bit on both, but what's important is the integration of these two pathways. Let's look at innate immune system for the next 5 minutes. When an infection or a possible infection is experienced by a newborn, let's say somebody cuts the umbilical cord, maybe the scissors were not very well sterilized, there's a little bacteria, that bacteria will enter the circulation, but child may get a little fever, leukocytosis, but generally has something, but they recover because they do have white cells and monocytes, and they complement system, so they can eat a few bugs which entered and kill them. So we are blessed with the innate immune system. If for some reason the innate immune system is overwhelmed, there's too much of bacteremia, then the infection gives rise to a disease, and then the doctors can treat the infection and the child will survive, or the neonate. If not, of course, unfortunately, the child may succumb. But more often than not, it's either subclinical infection or clinical disease. And when they recover, this battle was not fought in vain. The bacterial antigens, the complexity of the sugars, the carbohydrates, the peptides, the viral proteins is now sampled by the T and the B cells, the adaptive immune system. And we'll talk about how they sample, how they talk to one another, and they end up with what's called specific immunological response and with memory. So if the child were to see the same bug the next time, it doesn't take 2 weeks. In 48 hours, cells can multiply, make cytokines, kill the bacteria or the virus. With the result, we get no disease. And this is the basis of subclinical infection, which is what they think happens in daycare centers when you have lots of siblings, or by vaccination. Vaccination has to be done prudently, not haphazardly. So what I want you to appreciate is that the innate immune system does a job, but its very important job is even when it clears the bug, it hands over the message, the experience to the next generation of adaptive immune system. So you can see how there's a two-way street. And this two-way street is shown in this diagram, which I had taken from Abbas' textbook. When a microbe enters, there's epithelial cells, skin, GI tract, which block it. If they escape, the phagocytes eat it. If the phagocytes can't kill, it recruits complement to coat like sugar and then eat and destroy it. And then we have the NK cells and the innate lymphoid cells, which take care of the rest of the problem. So 9 out of 10 times innate immunity takes care of it. If for some reason it does not, then it hands over the antigen to the T and the B cells, and they divide, and we'll talk about the different kinds of T cells and B cells. B cells make antibodies, hence called humoral immune system. T cells make cytokines, recruit other cells, create a constellation of cells which together surround the pathogen and get rid of it. Also, the way we kill tumor cells. The beauty is innate system is very quick acting. It takes hours to get rid of it. If not, it hands over to the adaptive immune system. The adaptive immune system, the first time it may take 7 to 10 days. Second encounter, it's a matter of hours. Gets educated and retains that memory. So here's another way of looking at it. The innate immune system is seen in the purple, and the adaptive immune system is by the red line. Look at the innate immune system. There are different bugs, bacteria, virus, and fungi. Fungi have sugars, so they have to recognize the sugar like mannose. Bacteria have lipopolysaccharides, so they have to be recognized. Viruses have glycoproteins and glycolipids and peptides. So different invaders come with different weapons. And the body has what are called pattern recognition receptors, like toll-like receptors, nod-like receptors, retinoic acid-like receptors, and then it also has cytokines. So they can sample the pathogen based on the surface of the bacteria, not the individual bacteria, the constellation. It can say, I recognize it's PK because he's wearing a nice gray pant, and I think looking from here, it's a black or dark blue shirt. I can recognize Dr. Gupta because he's got a 3-piece suit, and which is fine. So I may not know the details, but I can recognize them from a distance. That's what innate immune system does, pattern recognition. Once it does it, it gets rid of it and then handles the fine peptides like pneumococci. Polysaccharide has 23 to 30 antigens. So the adaptive immune system makes antibody or T cells. Each of these distinct antigens, they are called epitopes. So you can see one is broad-based, like reading the chapter for the first time, get that theme. The second, adaptive, is detailed analysis. And the important concept of any immune response, T cells or B cells, is they come as naive T and B cells, they recognize the antigen, they grow, they divide, they make more of the same cells, and then they differentiate depending on whether it's an allergen or a virus or a tumor, and you get different kinds of effector T cells and B cells. We'll talk about that. Then they get rid of the pathogen, and once the job is done, you see they downregulate and they go into a steady state. It's exactly like what you do when you get 10 emails. You respond to the 10 emails, you answer it. Different people need different responses. Then after that, you shut the program and you go away. But your email still has a memory of what you sent. And apoptosis is like closing it down. It's programmed still there. So immune system should respond, will respond when it's intact, reach different levels of sophistication or differentiation, perform the various effector cells, and then it should shut off. But it keeps a memory, just like all the emails we get, it's in our old box. The other very important concept, which I'll make it brief, is specificity. An immune response to an antigen A remains an immune response to antigen A, and it will not react with an antigen B if they are dissimilar. In this diagram, again taken from a bus, they are showing two antigens, X and Y. You see, X is the purple one. When a host meets a pneumococci, it makes primary pneumococcal response. After it's cleared, it goes away, keeps memory to pneumococci. Next time it gets an infection from Staphylococcus aureus due to, say, atopic eczema, a scratch, it gets in. Now, staph is different from pneumococci. Staph is like the green box. So you get a staph response for the first time. And if it were also to see pneumococci, then that will respond even more vigorously. So the primary response is brief, short-lived, and leaves a memory response. The secondary response is very abrupt, very sustained, reaches a higher peak, and ends up in a memory response at a higher level. So every time you read a chapter, your memory response goes up, which is one of the reasons that many of us, read something, it sounds very difficult, close the book, forget, go away, come back, read it 2 days later, you understand a little more. Read it a week later, you understand a lot more. So this is all boosting up understanding and opening up neuronal pathways. Similarly, the immune system is opening up immune cytokine mediated function. Lots of similarity. So now let's look at what happens when an antigen enters the body. When an antigen enters the body, it has to be recognized. And the cells that recognize this are called antigen processing and presenting cell. The best example is the dendrite, and this is a cover picture from a science journal. Shows the dendrite with its the cell body with the dendrite sticking out, sampling the diplococci which is sitting there. It attaches, then it sweeps it inside, and then it eats it up. So the bacteria is eaten, as you can see on the right side. It is degraded by enzymes, and the antigens are then assembled with self-proteins in the phagolysosome, and the self-recognition molecules are human histocompatibility complex or HLA antigens. These are of two flavors, HLA class 1 and class 2. We'll talk about it very briefly. And then the disassembled bacterial or viral peptides are put on HLA class 1, and then the dendrite gives it to the T cell by cognate interaction. So what the dendritic cell or macrophage or Langerhans cells in the skin or the glial cells in the brain or the mesangial cells in the kidney, they're all examples of antigen processing and presenting cells. These cells pick up the foreign, put the foreign on the cradle of self-antigen and give it to their T cell and saying, "Yep, I'm presenting to you the invader. And as an example, this is our own molecule. So you see, the invader is different from self-antigen. So please respond to the non-self and don't respond to the self." So that's the concept of antigen processing and presentation. Now, this can be done at two levels. One is innate immunity. In innate immunity, as I already told you, the cell doesn't recognize individual peptide. It recognizes carbohydrates, sugars, lipids. And so they're called pathogen-associated molecular patterns or damage-associated molecular patterns. Like when there's a burns, when there's hot water which is spilt on our skin, the epithelial cells die and they're damaged. The body recognizes foreign, clears the damaged cell, so new epithelium can grow. So innate immunity recognizes broad structures. It recognizes the group, but not the individual. Whereas in adaptive immunity on the right side goes for very distinct epitopes, the types of polysaccharides. The receptors for the innate immune system is coded in a germline. So we have it at birth. They're like the toll receptors, Nod-like receptors, Mannose receptors. Whereas the receptors for the adaptive immune system, whether it's a T cell receptor or the B cell, can undergo evolutionary changes. They come like, for example, tomorrow PK may walk in with the red shirt and the gray pant. So he can change. And day after tomorrow, he may come with a khaki pant and a white shirt. So my choice of clothes may not be perfect, but what I'm saying is you can change it. And that's adaptive immune system. Innate, it's the same. Like you see in Many schools will have a uniform, so all the kids look alike. So, oh, sorry. So what I want you to see is innate recognizes broad strokes, adaptive fine details. And in order to recognize broad strokes, there's a variety of toll receptors. I just want you to look at the flavor, but don't get lost in the details. For example, if it's fungi, TLR, toll-like receptor 1 and 2, recognize it. It's a different fungi like Borrelia burgdorferi, Lyme disease. It recognizes outer surface protein. If it's yeast, recognizes what we call zymosan-like. If it's a double-stranded DNA from a virus, pathogens, it will recognize, or lipopolysaccharides like gram-negative bacteria, flagella like salmonella, or viruses like flu viruses, or DNA virus like herpes simplex. So all the aliens have different receptors, so they recognize the pattern. It is the same receptor which also recognizes self-antigen. Our own membrane messenger RNA, our heat shock protein, or our double-stranded DNA are also recognized by these toll receptors. On the top, when the toll receptors recognize them, they undergo restraint. They say, hey, this is self pattern. I recognize this. I'm not going to respond. They get a free passage through security. But if the pathogens come, the security is going to block. The toll receptors communicates within the cells, tells the innate cell, please process this miscreant and hand them over to the authorities. So that's basically what it does. And we have to protect at two levels. One is bacteria can come from the outside world, but the virus, they're very tricky. Like take respiratory syncytial virus. It can bind to an adhesion molecule inside the cells, multiplies inside the cells. But nature is smart. When they multiply and they release the viral receptors inside, then it can bind the virus. If it's coming from outside, the receptors outside. And here is a picture. TLR1 and 2, TLR4 are all outside. The bacteria bind, they're anchored to the membrane. These purple, what looks oval molecules send signals, what's called transcription factors to the cell. But if it's inside the cell, look at this, double-stranded viruses, single-stranded RNA virus like flu, single-stranded RNA viruses of a different structure, or viruses which don't have CpG residues. They're all inside the cell. They multiply, these toll-like receptor or in vesicles, they can capture the virus and then tell the cell, make an inflammation, get rid of that. So we can protect from the outside world as well as our internal milieu. Now that is innate immune system. What about adaptive immune system? Let's say a virus that enters my cells. Let's say it's a common cold, respiratory virus or adenovirus. The virus goes into the cytosol because it uses the adhesion molecules as a surrogate, like a false channel, binds to it, and the cell takes it in. Because it has a false passport, it's recognized initially as a masquerader. But once it goes into the cytosol, the virus multiplies. But don't worry, when it sheds its particles, the particles have to be gone through what is called a proteasome. The proteasome says these proteins don't belong to my body. So it breaks it up, chops it up, puts these protein peptides into a molecule called transport associated protein. So here the miscreant is captured by the officer in this airport. It takes it to the authorities. The authorities say, okay, now look, this is alien. This guy got a false passport. His fingerprints don't match. Therefore, this is his original identity. This is his assumed identity. So there's foreign, which you see in this dark little what looks like the toothpicks, and the other complicated molecules, HLA. So it takes it through the endoplasmic reticulum and then puts it into the endosomes. And the endosomes say, all right, I'm going to send this out as a miscreant with my self copy. And the T cell says, I'm the CD8 cell and I recognize HLA class 1. So I'm going to bind, I will interact and make perforance, granzymes, and get rid of this guy. But he is not, should not be there. So that's intracellular trafficking. But if the pathogen came from the outside world, you see that it binds to the cell membrane, it's phagocytosed, digested in the phagocytic lysosomes, which have enzymes, again breaks it up into different pieces. It takes it to the endoplasmic reticulum, and in the endoplasmic reticulum, we are making HLA class 2, and the class 2 has a piece called the invariant piece, which keeps it in position. So when the peptide fits in, the little pink spacer is kicked out, and now it presents it on the APC, and the CD4 cell says, I like class 2, I recognize class 2, so I will engage in this. CD8 cell on the top engages with class 1. So another dogma in immunology is HLA class 1 talks to CD8 cells, HLA class 2 talks to CD4 cells. So depending on the nature of the antigen and its presentation, it could either be a T cell CD4 pathway or a CD8 pathway. So this is how adaptive immune system works. Once it works, the T cell says, okay, what do I do with this? And the T cells are developing in the thymus. Initially, they're called double negative. They are 4 negative, 8 negative. They don't have a T cell receptor, but they are CD3 positive. As the T cell matures, it acquires CD4, CD8 T cell receptor. If the maturing thymocytes don't recognize anything, in other words, this is like reading a chapter in immunology to a kindergarten kid. He doesn't understand immunology, but one day it will be a professor. So that kid will say, it doesn't interest me. But if somebody tells a fairy tale, then the kid will engage. So if you don't recognize it, the cell dies by neglect. But if it recognizes too much, it's precautious. Suppose you have a 5th grader who understands immunology. This kid is super smart. They have to go through advanced training early and end up in medical school by the age of 16. And there are some wizards like that. So they can't be in the regular classroom, they have to be nurtured. But those that are properly programmed, if they recognize class 1, they become CD8 cells. If they recognize class 2, they'll become CD4 cells. If these cells don't recognize anything, then they become T regulatory cells. They say, all right, at this stage, I have not been recruited. I'm neither going into medicine, nor I'm going to engineering. I'm going to study a little more. And maybe I'll do bioengineering with a PhD, MD, whatever. So those cells become Tregs. They are not yet specialized, but they can act as a controlled homeostatic cell. So you can see how you can get different kinds of T cells. The important thing is this education takes place in the thymus as the thymus is developing. Once the thymus is gone, people have different views. Some people think there's still rudimentary thymus. Others think the skin T cells can do the job. We don't know who functions in adult, but there is the equivalent of thymic cells. And these thymic cells see an enzyme, a transcription factor called autoimmune regulatory element. What this autoimmune regulatory element says is, if it sees thyroid hormones, it says, okay, this is self, please don't respond. If it sees insulin, it don't respond. If this autoimmune regulatory element is mutant, then you end up getting immune disorders because you don't have the sensor pathway. All this is happening at the level of the T cell, and the similar thing is also happening in the B cell. So let's develop the T cell towards allergic disease. So the double positive cell can either become NKT cell or CD8 or CD4. I just want to introduce the different CD4 cells, and I'm sure you have got this in 2 of the lectures, but I may give you a little more details, hopefully, to add on. So this is called pleiotropism. What PK gave and the professor gave, we'll add on to. So the CD4 cells can differentiate different types of T helper cells. They're all CD4. One is called TH1, other is called TH2. Then we have TH17, we have TH9. We'll talk about more cells. Different differentiations, like cardiologist. He's a heart failure specialist. He's evoked potentials and electrical physiologist. This guy does only valvular heart disease. So the T cells have different subspecialization. If nothing happens, they become Tregs. The Tregs can either be generated in the thymus, they're called natural Tregs, or they can be induced in the periphery by cytokines like IL-10 and TGF-beta. When you give allergy shots, what you're essentially doing is you're stimulating induced T regulatory cells. And these Tregs go and tell the Th2 cells, which are making IgE through the B cell, and says, please shut off. We don't want you. You're working to excessively causing disease. So it is the induced Tregs which are very important. The natural Tregs help us to prevent autoimmune diathesis in the human being. So they work at different compartments, but they do the same job or similar job. So now I'm going to switch gears to B cells. B cells are very similar. They are born in the bone marrow. They are constantly developing through cytokines. They go through various stages like a kid, pre-kindergarten, kindergarten, elementary school, middle school, till they come to high school. So till that, automatically they mature. At that stage, after high school, they have to make a career decision. They want to go engineering, medicine, economics, social studies. So as the B cell matures, it makes immunoglobulins inside the cytosol, puts it on the membrane, and all mature B cells carry IgM and IgD on the membrane. So this is called antigen independent maturation. Once a pathogen comes, the pathogen will bind to this IgM, and depending on the pathogen and the cytokine, it will become IgG producing B cell or IgA or IgE. So that's called antigen dependent maturation, which I'll come back to later. So let's go back to the T cell. So analogy is T cells develop in the thymus and come out, B cells die. How are T cells activated? There are 4 signals through which the T cells communicate with the antigen presenting cell. I'll enumerate them first and then explain. Signal 1 is, I already told you, remember when antigen comes, it's chopped up, put on the MHC class 1 or class 2, and it presents to the T cell. The antenna on the T cell which recognizes this is called the T cell receptor. So APC T cell receptor communication cognate is signal 1. Signal 2 is when the T cell receptor is stimulated. For example, you turn on your TV, you get music, but you then have to tune the channel. Do you want news? Do you want the comedy? Or do you want the drama series? So for the T cell to respond, it has to be activated, and they are called co-stimulatory molecules. You will say, all right, keep going. Dr. Gupta, is it breaking up? Are we okay? Is it all right?
इसको लेके बीच वाली सीट पे बैठ जा इसको लेके माइक
Okay. Okay, shall I continue?
हाँ धीरे बोलो ऑनलाइन ना जो
PK, shall I continue?
बता दीजिएगा मैं भी चल पड़ूंगा आपके साथ बताने के लिए
कोई कर लो
Yes, Vinay? Okay? All right. All right, okay. So, signal 1.
Dr. Murali,
Yes,
I am audible to you?
you're audible to me.
Yeah,
I was
it
getting
is.
a lot of noises, so I was not sure whether the communication was okay.
It was a little breaking up. Can you restart from the 4 signals which you were starting enumerating from signal 1?
Okay, let me go back.
Yeah, please.
So,
Thank you.
So, when antigen is presenting by an APC to a T cell, the first signal is the APC presents antigen to what's called the T cell receptor. The T cell receptor is like an antenna. So, and I already told you the antigens are either broken down intracellular pathway, which is MHC class 1, or extracellular pathway, which is MHC class 2. So this antigen is given to the T cell receptor, and the T cell by cognate interaction recognizes signal 1. Then the T cell has to make a big decision. Do I continue with the immune process or do I stop? Is this a false phone call that's coming, or is this a true phone call that's coming? So in order to make this procedure go on, the T cells are looking for another signal and saying, yes, this telephone number that you're receiving a call is from PK, very important, please take it. So that's called co-stimulatory molecule. On the other hand, if it's what they call the slammers, the telemarketers bothering you all the time, you say, that 800 number, I don't recognize it. It's a nuisance. I'm not going to pick up the phone. Let the message go through. So there are times you have to respond to signal 1, and there are times you don't have to respond, or you should not respond to signal 1. So these are called co-stimulatory or regulatory molecules. Very, very important. The co-stimulatory molecule are CD28, I'll show you. The regulatory molecules in normal physiology is called CTLA4, and in tumor immunology, it's called program death 1 and 2, and we'll show that in a minute. Then, once the T cell says, okay, I got signal 1, I've got the antigen, I got the go signal, I should respond, that cell will now make cytokines. And the cytokines it can make depends on the antigen. If it is a bacteria, virus, and fungi, it will make IL-2, IL-6, and activate the inflammation. If it's a self-antigen, it will say, oh no, we should not respond to this. This is my own double-stranded DNA. It's been erroneously processed and presented, and it makes TGF-beta and IL-10, opens up the Tregs, and the Tregs, the buck stops here. I don't want a lupus, or I don't want an allergic disease. And what's very important in the future, we don't have full answers, though it's coming slowly, is the microbiota is very, very important. And there's some beautiful papers about butyrate in children who don't have allergy, whereas there's less butyrate in children who have food allergy. So the microbiota may influence it. So we always used to say signal 1, signal 2, the 3. Now we should include signal 4, even though we don't have hardcore data on that pathway. So let's look at signal 1. Here is the antigen presenting cell on the right side. This is the MHC molecule. The antigen peptide is presented in the cradle of the MHC to the T cell receptor. The T cell receptor has alpha beta chains, gamma delta, and it has got sites where it can create energy or phosphorylation. So that's on the zeta chain or the theta chain, which you see here. So it's ready to go, but it will not go unless it receives the OK. And the OK is your CD28. The antigen presenting cell has 2 molecules called CD80 and 86. For those like PK and me, we learned it as B7 and B7.1, but nowadays the next generation will learn it as CD80, 86. The yellow one engages CD28. Look at what it does. It gives more energy. It gives more PA3 kinase. This PA3 kinase with the phosphatase will then activate the cell, and the cell will start making cytokines. On the other hand, let's say the antigen is cleared out, the job is done, there's no antigen, then the same CD8086 will bind with CTLA4, which is like a brake. You've driven the car, you reach your destination, you put on the brakes, put on the parking brakes, turn off the engine. So when the job is done, the CTLA4 is activated, and CTLA4 has what's called sulfhydryl phosphatases, which turn off the pathway, so the T cell goes into quiescent stage or becomes a memory T cell. So this is signal 1, antigen T cell receptor. Signal 2 is co-stimulatory or inhibitory as CTLA4. And showing you the same important concept. APC, antigen presentation T cell receptor. If 8086 engages CD28, this naive T cell will proliferate. It will make interleukin 2, which is autocrine, activates the cell, so you get more T cells of the same antigen specificity. That's why if you put a PPD and you get a positive response, that means you're recruiting T cells which recognize PPD and they proliferate, call in other cells, and you get a granuloma at the local site. If you don't have memory and if you didn't recognize antigen, nothing happens. So you're testing T cell access. Now, what is important now is we talked about signal 1 and signal 2. Signal 3 are cytokines and signal 4. But there's one other important thing. If it is a tumor cell, you killed it with chemotherapy, but the tumor cells are very nasty. They make new antigens and they're called PDL ligand. They activate the programmed cell death and they try to kill the host T cells. So the tumor cell is trying to overcome the immune system. So what biologists did is, okay, you block this pathway. Only if you can get rid of this pathway, then the T cell can function and kill the tumor cell. So what we do is we use antibodies, monoclonal antibodies, developed in the test tube to block it. And for example, there are two drugs called nivolumab, ipilimumab, pembrolizumab, which block PD-1. So the inhibitory pathway, the one that is acting as a mischief pathway, is blocked. Or it can block the PD-1 ligand, and that's called atezolizumab, which blocks the ligand. So these three drugs have changed treatment of metastatic melanoma, breast carcinoma, etc. So here you want to activate the immune system working on the second signal, which is co-stimulatory molecule, which are bad molecules. You want to stimulate it with CD28. So what I have done is I've given you signal 1, signal 2, including PD ligand. Signal 3, we'll talk about cytokines. Well, cytokines are very pleiotropic. When an antigen binds to antibody or on the B cell can also present antigen, it's taken in. See, the antigen is given on HLA class 2. I'm repeating a little bit. TCR is activated. Co-stimulatory molecules are engaged. So you get a lot of energy. The cell makes a variety of cytokines. IL-2, 4, 7, 9, 15, etc. And these cytokines bind to the receptor and activate the B cell. For example, if 4, 7, 9, and 15 bind, those B cells will start making IgE. If the cell makes gamma interferon, and if gamma interferon binds, they make IgM and IgG. So you see how gamma interferon is more protective humoral immunity. IL-4, IL-13, IL-9, it makes Th2 response or allergic response. So the cytokine influences the milieu. And that is what I'm going to show you by this beautiful slide, which I've taken from Middleton's latest textbook in allergic disease. This slide is the best I can find in the field. So I'm going to spend a couple of minutes because this is the modern immunology. So antigen presenting cell, we talked about it, innate adaptive, presents it to the naive CD4 cell. If this cell recognizes the antigen structure and makes IL-12, it activates what's called a transcription factor, which tells the nucleus of the cell, please make gamma interferon. Gamma interferon will kill viruses and make IgM and IgG, make humoral immunity. So this is called the Th1 or T helper cell type 1. If the naive T cell says, no, I don't see it as a bacterial peptide, I see this as a food protein, or I see it as parthenium, or I see it as a dust mite, it processes it and tells the T cell says, I'm going to make IL-2, IL-4, and IL-7, IL-9, etc., and give it to the Th2 cell, which will be a different responder. Its transcription factor is GATA3. Don't worry about transcription factors. And that will make different cytokines. 4, 13, which will cause IgE production and will bind to extracellular parasites or pollen grains. On the other hand, if it sees pathogens which are coming through the GI tract, it makes IL-4 and TGF-beta. TGF-beta is mucosal immunity, stimulates Tregs, and makes IgA, which is a mucosal antibody. And then it tells another T cell called Th9 to make IL-9, and this destroys helminths and how we don't react to milk proteins. So you can see Th1, Th2, Th9. If the naive T cell receives signals and makes IL-6 and IL-21, it's the new cell called the Th17. It has a different transcription factor called ROR gamma T, and it makes IL-17, just remember that. And IL-17 brings in neutrophils and monocytes and kills bugs. So asthma is a syndrome. If it is initiated by pollens and dust mite, it goes via Th2 pathway. But if asthma is triggered by ozone, particulates, tobacco smoke, it goes through Th17, so you get neutrophilic asthma, whereas Th2 is eosinophilic asthma. So you can see how you get different endotypes of asthma. Then, very important, if the antigen is sequestered in the lymph node, and the lymph node says, yep, this is an intracellular pathogen, it's also a bacterial pathogen, it stimulates T follicular helper cells, and that makes IL-21, that's the chief one, and that activates B cell to make IgM to G to A to D. If there is no proper recognition, if there's nothing happening, then the stroma says, I always have IL-2, I always have TGF-beta, so I don't see a pathogen, a parasite, a pollen, so it develops into Tregs, and Tregs make IL-10, and IL-10 is immunosuppressive. When we give allergy shots, the aqueous allergy shots or oral immunotherapy, those antigens are processed by the Tregs, and the Tregs make IL-10, and the IL-10 turns off the Th2 response. So you see there's a lot of communication here. The developmental pathways is determined by cytokines upstream, or I'll call it the afferent pathway, then like the neuron, and then this is the efferent cytokines. It's a great slide. The more time you spend, the more you appreciate human diseases. For example, ankylosing spondylitis is Th17. Rheumatoid arthritis, Crohn's disease is Th1. Ulcerative colitis is both Th1 and Th17. Multiple sclerosis is Th1 and Th17. Mantle cell lymphoma, diffuse follicular B-cell lymphoma is T follicular driven pathway. So you can take each cell and enumerate the diseases. Microbiota, I'm not going to say, but there's good and bad. But the concept is when we have microbiota, the good saprophytic microbiota, they shed antigens sampled by toll receptors. The toll receptors educate, innate, adaptive. We make protective IgG and IgA, and we live in a symbiotic relationship with bacteria, and we don't get allergy. On the other hand, if the microbiota are not there or they're disorganized, you give too much antibiotics, then you don't get innate stimulation. So the adaptive cell has no education. It simply makes IgE, and you end up with allergic disease. So this is the whole area that needs understanding: prebiotics, probiotics, etc. So that's the 4 signal. So I've summarized these 4 main important ones. I've not shown you Th9 and T follicular cells, but just understand that cytokines cause the differentiation and development of these T cells, which in turn make interferon, and Th1 disease is important in protecting us against TB and fungi. If there's deficiency, you end up getting disease like HIV and primary immunodeficiency. If it's too much Th1, then you get sarcoid granuloma. Th2 is good and bad. The good is it protects against parasites. The bad is it causes allergic disease, which is why you're studying how to control with antigen recognition, antigen avoidance, pharmacology, and immunotherapy. Those are the hallmarks. Then neutrophils. This is the big point. Not all asthma is eosinophilic. That's why drugs like mepolizumab, benralizumab, mepolizumab don't work in all asthma. They work only for eosinophilic asthma on this area, not here. We don't have good drugs in this pathway, and steroids don't work as well. So you remove the antigen, the ozone, the tobacco smoke, the pollutants. Inflammatory bowel disease, psoriasis, there's a whole group of monoclonal antibodies like ikizumab, rizolizumab, which all block IL-17. Then TGF-beta is the good guy. It provides mucosal immunity, programs regulatory cells, and if you have a deficiency, it causes a syndrome called IPEX, immunodeficiency, polyendocrinopathy, eczema skin, and it's X-linked. So they have a multi-organ autoimmune inflammation. So this, I think, will be a useful way for you to put things together. Hypersensitivity pneumonitis involves Th1, Th17, CD8 cells, NK cells, and immune complexes. So that one disease, whether it's acute, subacute, or chronic, uses different pathways. So I'm going to quickly end with the little B cell biology. I told you this is like a kid going to elementary school, coming out of high school. After that, it depends on how the kid faces a mentor. If it comes in contact with Vinay Mehta, Dr. Niraj Gupta, and the kid sees them in their office, they're doing some excellent work, and that kid will say, I want to study biology, I want to go to medical school, I want to become an allergist, immunologist, so I can also help other people. So it depends on the mentor. Or if it sees a great engineer, wants to build cars, planes, bridges. So again, antigen presentation T cell. Here the co-stimulatory molecule between T and B cell is CD28, but there's also another one called CD40, 40 ligand, and that with cytokines causes class switch. And that class switch is what gives rise to immunoglobulins, whose structure you know very well, and you get initially IgM, which is intravascular, fixes complement and kills bugs. So bacteremia is fought by IgM. If the bug escapes into the alveolar space or the kidney space, M can't get in, it's too fat. IgG is a class switch. IgG will track it. If it is an allergen, it will bind to the mast cells. So when the allergen binds to the mast cells are activated, releases mediators, you cough up the allergen. So cough is a protective reflex, trying to get rid of the pollen. If you have a parasite in the gut and you get abdominal pain and diarrhea, the mast cells are causing intestinal motility to wash off the pathogen. That's the way it protects it. But if the parasites or the bug hides in the GI tract, then IgA and TGF-beta take care of it. So different classes fight the battle: intravascular, interstitial, cell membranes, mucosal surfaces, or in the mucosal blanket. And how do antibodies work? You know that very well. So I'll make it fast. They can neutralize toxins. They act as a scaffold to coat bacteria. And by doing that, they engage Fc receptors. They're eaten up. Then they recruit complement to activate inflammation and punch holes. So you get various properties. And again, like a T cell, the B cells are activated. They expand clonally. Then with cytokines, they become different kinds of, they become different kinds of B cells, and you leave a copy as a memory B cell like a T cell. So there's a lot of similarity between the two. I know time won't permit me to go into complement, but complement synergizes with IgM and IgG, activates a series of proteins which release products which cause inflammation. They coat bacteria, that's called opsonization, which is a preliminary for eating up the bug. Then the terminal pathway will activate the membrane and punch holes and the bacteria or the virus will die. So both pro-inflammatory and membrane lysis. And that's what happens to the membrane. You get holes and the bug dies. Complement also attracts neutrophils. And we are not talking about it, but you will have to know neutrophils circulate. They attach to the endothelium. They come out. Then they eat bacteria and they activate a very important superoxide hydrogen peroxide pathway. It's called the phagocytic oxidase. And they generate superoxide, hydrogen peroxide. They combine with the halide to form bleaching powder, and that's how cells kill bacteria they eat. And if you don't have this, then you get chronic infections, and there are diseases called leukocyte adhesion deficiency syndromes, Chediak, Higashi, etc., where the neutrophils don't work. So I'm going to sort of end this talk integrating something we know and many things we don't know. Asthma is a complex trait. It's not one disease. There are a number of genes. But it's not just the genes. the genes interact with the environment. That's why people who have the genetic predisposition come to the USA and they see ragweed, they get ragweed allergy. But then they say, but they had the genes, but they didn't have the environment of ragweed, though Parthenium cross reacts. So a naive individual, given the genetic predisposition, which is on many chromosomes, can either be normal with no asthma, where you get protection, or they can develop asthma. What are the factors which protect? These have been studied. You know, the hygiene hypothesis says if you have early bacterial exposure, you develop innate adaptive immunity, the Th1 pathway. If you don't, by default, it becomes Th2. And the examples, if you're in daycare, you get subtle subclinical infections, but you educate Th1. If you have older siblings, they get infection, they bring it home. Certain ailments in the old world, hepatitis A is very common. There, the asthma prevalence is less because the virus educates the innate immunity. People living in farms are exposed to fungi, good endotoxin, good bacteria, and they develop Th1 more than Th2. So there are what are called epigenetic. They are not in the gene, but they regulate gene function. Then there are bad epigenetic factors. Mothers who smoke, pollution, absence of germ-free environment, a kid who gets antibiotic for everything. The nanny uses gloves to handle the child. Child never goes out and plays, never falls and scratches the knee and gets infection. Only child syndrome, no siblings. Then they protect it. As soon as they catch a cold, they get antibiotics. So they kill the good bacteria in the gut. And then vaccination has been put epidemiologically, but there's no good data on that. Low lactobacillus is bad. Good butyrate is very good on that side. So in a modern society, too much of shielding has ended up in changing the epigenetics, and that's felt, though not conclusively, to promote the Th2 phenotype. So we are trying to get some of the good old world habits. So the same immune system which we taught, when they are blocked at different levels, they don't develop, you get primary immunodeficiency. So you can take the same talk and turn it to immunodeficiency, allergy, transplant rejection, and tumor immunology. So what I have done, hopefully, is talk to you about how the immune system is structured and how they function. This picture is historic. If you come to Boston, you should come and visit us. This is the original MGH hospital. It's called the Bullfing. It's still there, but it's surrounded by new buildings. The immunology lab where I'm fortunate to work is in the historic building. It's where the lab started, it still is. And what I want to do is to thank you folks. And here is a building called the Ether Dome, which is next to my lab. And that's where there's an auditorium. And that used to be a surgical theater where they first showed ether as a useful anesthesia, and you can operate human beings without pain. So they have a little museum, which is very interesting. Every anesthesiologist who comes takes pictures, they enjoy it because that's how their field began. So there's a lot of history, but I just thought I'd share it with you since you gave me the opportunity to share some time with you. So what I would like you to do is, Dr. Gupta and PK and Vinay and company, the slides are available. So take a look. And if there's some area that bothers you, you can get my email. PK will give it to you. It's murali50@aol.com. And you can shoot a question or two. I'll respond hopefully within 2-3 days. Sometimes there are lots of emails. I wait for the weekend to catch up. And immunology is fun, but it is expanding rapidly. But you can say the same thing with cardiology, the same thing with GI. But immunology is necessary for understanding GI, hematology, rheumatology, allergy, tumor oncology, now even cardiologists and neurologists, because many of the neurological syndromes are mediated by autoantibodies. But the fundamental is what we have talked today. So I'm going to stop here and questions, comments, and Niraj, you wanted a little extra time. I have the time, and your friends
Thank
and
you very much.
family.
It was one of the best talk. I was fortunate to hear you for the second time now this time. So I will straight away go to the students and any question, please interact with Dr. Murli.
And don't feel shy. No question is stupid. Every question is
Anything,
good.
please. Any question related to immunology, allergy you might feel, please ask. Yeah, please take this. Dr. Murli, Dr. Gorika is in touch. She will be asking.
Okay, with pleasure.
Hello, sir. Good evening. So
Good
my
evening.
question is that during the initial part, we say during infancy, if the baby is exposed to a lot of infections, it leads to the Th1 pathway. So they're prevented against allergies. But as when they become sensitized or when they're, suppose there's a history of atopy, and in case they catch infections once they grow up, in that case, we say the allergy worsens. All viral colds travel down to the chest in atopic patients. So in that case, why is the Th2 pathway activated when these kids get infections later on in life?
I think it's a great question. I'm going to repeat the question. Question is, I presented evidence that Th1 stimulation is anti-allergic. On the other hand, a child who's atopic, let's say has asthma, gets a viral infection, that infection doesn't protect them, that makes their asthma worse. Why is this funny thing happening? So it depends on the pathogen. The bacterial infections, fungal infections induce Th1 response. Viruses are very tricky. Initially, when the first time they come in, they will get inside the cell, work through the TLR, and they get eliminated. So you never make a good antiviral immunity because they're not exposed. That's why you keep getting influenza all the time. They change their epitopes. They don't bring a good immune response. Unless you take the antigens of the influenza, couple it with what's called activators, like Freund's adjuvant, and that's how the vaccine is made. So you boost the T cell, B cell communication. So bacteria are protective. Viruses are not protective by themselves. Then in an atopic child, there are two things happening. Whether it's atopic asthma, atopic dermatitis, or food allergy, there is a barrier defect. There's an epithelial defect. Normally, when viruses get into our respiratory system, they bind to the epithelium transported inside, and then we kill it using TLR. But in allergic disease, and as you know, in atopic dermatitis, there's a protein called filaggrin. They are all mutant. So the epithelial barrier is broken. So the virus goes directly into the deeper tissues. They stimulate the innate lymphoid cells, which make bad cytokines, like the IL-4, IL-13, and it makes the allergy worse. So, in other words, if you go out in the rain with a raincoat, you are protected. And that's what normal skin is, normal lung is. But if you don't have a raincoat and there's a torrential downpour, you go out, you will get wet. So, the viruses work very detrimentally, causing worsening asthma, rhinitis, food allergy, atopic dermatitis, and the barrier is broken. We know a lot about the skin barrier. In the lung, we are just understanding it. And the same thing is being understood with celiac disease. In celiac disease, why do gluten make it worse? Why doesn't it make? So they have a barrier defect. So the gluten peptides get into the GI tract, stimulate the immune system, and they make tissue transglutaminase antibody. And so there are people working on new drugs which cause the barrier. And Vinay Mehta will tell you there's ceramides in the skin. There are creams which, if you apply, acts as a barrier. So dust mite and other antigens will not get through the skin. So your question is very, very important. How is the virus seen by the body? Through intact anatomy, physiology, or through a deranged, inflamed mucosa? It's a great question.
Dr. Murali, it was a fantastic talk. It was eye-opener for probably many of us. Just one question. Nowadays, there's a lot of pre-pro antibiotics available, means probiotics available in the market. and somehow they are indiscriminately used by general public. We don't know the research, but recently we have seen around 3 cases of septic shock because of those, you can say, bacillus which has been used. So, what your research says, are all are helpful, or they can actually harm the patient, not only in allergy, otherwise also?
I think this is a great question. This is where the marketing has superseded science. So there is no question there is good microbiota and bad microbiota. And there is a nice journal of allergy and immunology 2 months ago which addresses microbiota. And where they've shown is, if you look at infants born to mothers and who don't have allergy, and you follow those infants, they make a lot of butyrate, which is made by good bacteria like Clostridium. On the other hand, and Bacteroides. On the other hand, children who get atopic disease, when they sample the bacteria, they are bad communities of bacterial colonies which make non-butyrate fatty acids. So we are still in the infancy of understanding. It's, I think, unfortunately, pre and probiotics have not shown any therapeutic benefit because each disease varies and each community varies. The microbiota in the Indian population on a vegetarian diet is different from the microbiota in an Indian population with a non-vegetarian diet, and is different from microbiota who eat a Mediterranean diet. So there is actually a very good clinical pathological conference in the New England Journal where a patient with ulcerative colitis and Crohn's disease read about probiotics, bought a compound which had pathogenic bacteria, took that probiotics, came with fever, diarrhea, and died. And when they found this bacteria, they opened up his probiotics at home, cultured it, it had the same species and the same genotype. So your point is well taken. I think we have to educate the public and say, eat a good balanced diet. Certainly include cheese, yogurt, leafy vegetables, fresh fruits. They all have good bacterial producing properties or stroma in which bacteria can grow. In terms of treatment, I think the marketing and the hype and saying, oh, I take probiotics every day, I take yogurt with probiotics is good, is more marketing than science. There is no evidence as yet. It's probably going to take a few decades before we get the full answer. So you're, I think that's where we need. Yes.
Just if there is a diverse microbiota,
Yeah.
it is protective. If it is just one, like a probiotic is not diversity. So is that one of the reasons why it is not effective?
So that it's a combination. Like everything else, you know, all the good cooks will tell you, you can add spices, you can add salt, you can add garam masala, you can add curry powder. That doesn't make a tasty dish. It's the right composition at the right time and how it's blended together. So the beauty is, yes, we are all polymicrobial. That's one. And it also depends on the diet we eat. It also depends on our own constitution. So I think marketing, you can't have one compound for the entire world. So that's where the whole thing arises. It's polymicrobial, it varies regionally, it varies with diet, and it also depends on what the basic microbiome is. You and I may not have the same microbiome. And it changes with diet. And that's what the latest study which I got from the Broad Institute is. They took human volunteers, put them on a vegetarian diet. The microbiota is different. Put them on red meats, it changes. Put them on a lacto-ovarian diet, it changes. So we need to know what is the basic diet, what is their baseline, and then you have to modify it. And that answer we do not have. So I would suggest the Jackie issue on microbiome addresses all these. And I'm hoping to introduce some of these in the chapter I have to give you a peek.
Thank you. Good evening, sir. Connected to the same question about the microbiota. As you said that the first three signaling pathways, 1, 2, and 3 we already know. And you said
Yes.
the fourth, which is joining in, is the microbiota. So in
Yes.
clinical practice, does that mean if the person or a patient of say asthma or some allergic or immunological or inflammatory disorder has been added microbiota as a therapy, would have a different response to the one who has not been added with that?
Okay, it's again a very good question. What you're saying is, everything being equal, will the addition of microbiota influence the outcome of an asthmatic patient? We don't have the data because we don't know which patient needs what kind of microbiota. So that's why I said it's a question mark. You can make animal models, you can study it, but as of now, we have no way of knowing which asthmatic will respond to what kind of microbial changes to bring a desired outcome. So till we know that, we should just stick to a good healthy diet, which is what everybody will tell you, a mixture of fibers, less sugar, uh, proteins in a balanced diet, and avoid as much of artificial compounds as you can, because nobody knows what they do. So I think microbiota we need to wait and watch. Just like, when PK and I were growing, people are arguing whether it's a T suppressor cell. Some people said yes, some people said no. It took 20 years to show that the T suppressor is actually the T regulatory cell, and now we know they have a value. Similarly, I think probably in a decade or even less, people will find out which disease, like psoriasis, what's the role of microbiome, what's in asthma, what's the role of microbiome in atopic dermatitis. It may not be the same, it may be different. So we don't have that knowledge at all. But I think it's important to keep reading about it without practicing it, but let the science develop. And then once there are clinical trials and well-documented pathways, then we can adopt.
Sir, I was having a little reading about this microbiota, and regarding the lung allergies or the respiratory allergies, there was one probiotic which I came across, lactobacillus GG. So this lactobacillus GG was, like, as per that reading or few of the articles, was found to having some protective effect. So is there any take regarding lactobacillus GG?
Okay, so there have been studies and looking at lactobacillus. There have been studies looking at bacteroides. I unfortunately don't have the slide. The problem is, in some studies it helps, in some studies it did not. So when they did a meta-analysis, what they found is lactobacillus is not a bad one. It may help, but it doesn't hurt. But as our colleague said, there are others like Clostridia, and there are like bacteroides. We have to be careful. They can cause disease. So those kind of probiotics, which have those, we have to avoid. And where is lactobacillus? It's found in yogurt, it's found in cheese, and it's found in India, of course, we use buttermilk. Here they don't. So that is all cultured good fungi that help. So I would say, tell them to eat a diet which contains lactobacillus. So they can have yogurt, they can have buttermilk, they can have cheese if they're not milk allergic. And so that will be useful, natural foods, rather than giving capsules and tablets, which cost a lot of money and we don't know. But lactobacillus is one of the promising ones.
So I have a question. If in a
Yes.
patient, the allergy and autoimmunity is coexistent, it means Th2 cell and Th17 both are activated. So shall we approach such patient differently in terms of choosing the pharmacotherapy, as probably the cytokine milieu is different, or immunotherapy, or maybe biologics, which should be the choice of the treatment, in which the allergic and rheumatology both are, rheumatic disease both are coexisting? Autoimmunity.
Great question. And I'm going to take a disease as an example, or maybe two diseases. Let's take allergic bronchopulmonary aspergillosis. Allergic bronchopulmonary aspergillosis has Th2 IgE pathway, but they also develop granuloma, infections, and bronchiectasis. So they have a Th17 excess as well as a Th1 excess. So what do you do here? When you have a Th1, Th2, and Th17, so there is no magic bullet of one or two cytokines. So the first thing is, who is the driver of this pathway? It's the Aspergillus fungi. So you try to decrease the colonization. So how do we do that? First, you can use steroids to block the IgE pathway, promote bronchial hygiene, treat the associated bacterial infections. When after this is done, since we don't know, we can't manipulate all the cytokines, because you block all of them, you'll make an immunodeficient patient. You'll make a patient who can't fight bacteria, who can't get rid of parasites. So we try to decrease the aspergillus, and that's where itraconazole and voriconazole come in the pathway. So you decrease the antigen, you minimize the immune response. So there you see, it's one, you still don't have a specific treatment, but you modulate the various axes of inflammation and eliminate or minimize the triggering antigen. The second disease I'll use is EGPA, eosinophilic granulomatosis with polyangiitis, the one we were all grown up as a Churg-Strauss syndrome. Look at Churg-Strauss syndrome. They have eosinophilia, they have asthma, and clearly they have Th2 elevated IgE, but they also form granuloma, so they have a Th1. So we have no idea what the antigen is in EGPA. So what do? If they have a lot of eosinophilia and wheezing and eosinophilic destruction, we can use steroids, which help a little bit. But as a steroid-sparing agent, nowadays they use IL-5 or IL-5 receptor antagonist. That's where your mepolizumab and benralizumab come in. But what do you do when they have granuloma? So then you may, the same steroids which help the IgE Th2 eosinophil inhibition also inhibits granuloma, which we know is the way we treat sarcoid, for example. So steroids actually turn off both Th1 and Th2, but there's a price to pay. The price is your osteoporosis, glaucoma, diabetes, etc. So what they're now trying to do is target it. And we know the main component is eosinophil, so choke it, block it, and see if the granuloma goes away. And in early studies, it seems to work very well in EGPA. So take lupus. All lupus is not the same. Sometimes they make too much antibody, so rituximab works very well. Sometimes it's not antibody, it's inflammation with IL-6 and TNF-alpha. So IL-6 is called tocilizumab, so use another monoclonal antibody. So the future is diseases have a name, lupus, but then within the lupus, there are different types, and they are called endotypes. Similarly with asthma, if it's a neutrophilic asthma, using dupilumab is not going to help. You have to find something else. We don't have any there. But if it's eosinophilic asthma, dupilumab helps. But if the asthma is due to, for example, atopic diathesis, allergens, the best thing is to identify the allergen and get rid of it or induce immunotherapy. That's why allergen immunotherapy, even though it looks old-fashioned, it's 100 years old, we don't know the mechanism, it's probably the safest modality without damaging, and definitely it's cost beneficial. So I given an answer to your question in a circuitous way, but you have to take each disease and dissect it.
Good evening, sir. There is
Yes.
a basic questions regarding with respect to asthma, sir. In the when we are taking the family history, maternal side, maternal portion is more concerned rather than paternal. What is the cause? What is the reason behind it? In one of your slide, you have shown there is a maternal smoking. That is the concern
Yes.
point. Is it
Yeah.
another thing also, or why we are concerned more with the maternal rather than paternal?
Right. It's a brilliant question. Actually, the question is, why is the maternal influence more than the paternal? And I had a slide where maternal smoking as a risk factor. See, this always bothered me many years. Unfortunately, there's a PhD, MD student working with me last month in immunology, whose PhD was mitochondrial DNA. So what he actually showed me his data, and there's a paper in Science that just came out, that the mitochondrial DNA, mitochondria is a high energy packet. It has its own DNA. That DNA is transmitted through the X chromosome. And it looks like mitochondrial DNA may carry messages for disease propensity. So based on his work, which was all done in animal models, that female animal models, their mitochondrial DNA is different from the father mouse. And when they look at the offspring, they're inheriting the maternal DNA as well as the mitochondrial DNA. And it's probably the mitochondrial damage from smoking, mitochondrial damage with pollution that's changing the epigenetics of the mother, and that is passed on to the baby. So that seems to be the logical explanation based on his animal model. There is no human studies as yet, but there is definitely DNA in our mitochondria, and that's inherited from the mother and not from the father. It's X chromosome linked. So it's very interesting how the genetics, the mom has a little better influence on the baby than the dad. Yes, PK. I'm not getting PK's question. The audio portion is not coming through.
Now it's audible, sir?
Yes, now it's audible. Yes,
Hopefully, the
PK.
last question. I know you need to get out.
No,
In
no.
the same context.
Yes.
Some reports of prophylactic immunotherapy, prenatal immunotherapy. You have any comments on that?
I will think it's an important area. I'll tell you why. I don't have any data. So what I'm saying is hypothesis based on biology. Let's say the mother is atopic. The mother is allergic to, say, parthenium. So the mother has sensitized T and B cells to parthenium, and those cells do go through the placenta to the baby and can tell the baby's immune system to make IgE. So that has been shown that IgE is not transmitted through the placenta, but the T and the B cells do go through. That's called maternal engrafting of the baby. Now, imagine you take that mother and you give her allergen immunotherapy, you decrease her Th2, you make more Tregs, so she'll pass that to the baby. So it's conceivable, and I'm being very cautious. I'm saying conceivable, possible. There's no data that by giving that, you can transfer more Tregs to the baby. So when the baby were to inhale parthenium or dust mite exposure, already starts off with a big bank account of protection. So the mother not only gives IgG, also can give Tregs to the baby. And that's why allergic mothers have propensity to have allergic babies is high. If you're non-atopic mother, then the children have less. So it is possible. Somebody has to do a clinical trial. You got to get a large numbers of people who are atopic, non-atopic. One gets immunotherapy, other does not. Show the immunotherapy worked in the mother, then follow the babies over the next 5-6 years. So that is going to be difficult, but a very important study. Vinay has a question.
Yeah, last question, just to provoke some discussion.
Yes.
What do you think about, are there potential disadvantages of the deworming programs that are instituted in developing countries? Do you think that deworming is contributing to the rise of inflammatory diseases in this part of the world?
Okay. So I think it's a very good question. Hard to answer it, but no doubt in the countries, what they call the developing nations, Ascaris lumbricoides is actually protective. And so there is one GI immunologist who took Ascaris suum ova, and he actually gave these to a bunch of asthmatics, right, monitoring the worm load, thinking if they get Ascaris, they'll have protective antigens, the asthma will get worse. The trial had to be stopped because there was no benefit at the end of 1 year. And a couple of people got back Ascaris overinfection. So the story is, we can only wait for epidemiology studies to come through. So let's say they do deworming of one village, follow what happens to the children going dead. Then you have no deworming in another village, and then see what happens very similar to the Philadelphia studies of the the population, which are very tight. So I can suggest we need to have epidemiological studies. It's very hard to say.
When we did the rural versus urban
Yeah.
sensitization published in JASI in 1997, we
Yes.
looked at the, we did not do any stools for OR parasites, but deworming was going on by the authorized by the government in all those villages. But the atopic sensitization was extremely low. It was not going up. But in the city, it was like 7 times higher.
Yeah. So I think what it is telling us is the natural infections with worms are protective, and in the city there are other epigenetic factors, which may be pollution, smoking, that is overriding the protective balance. Like hepatitis A is the best example. Hepatitis A is protective against atopy because the gene which makes them get hepatitis A is protective against asthma. So I think these are very hard unless you do large epidemiological studies, like the Hutterite. Remember in Philadelphia, born in farms, growing in farms with bacterial infection, they get less asthma. The same Hutterites coming to America and living outside the Philadelphia community, living like us, the asthma is high. So I think epigenetics has a lot to do with it. So I think both Vinay's question is very hard to answer. We need much better epidemiological studies.
So in the same context, my question was regarding the vaccination against hepatitis A or typhoid. So what is your take about the incidence of allergies in those patients when we are routinely vaccinating against hepatitis A and typhoid?
Okay. So I think there is data against hepatitis A. Hepatitis A is a bad disease, even though majority of us get the disease, have subclinical or clinical hepatocellular inflammation we become. There are people who go on to hepatic necrosis. So the advantages of hepatitis A vaccine outweighs the emergence of allergic disease. So the present thinking is vaccinations against hepatitis A is more beneficial than detrimental, looking at the population at large. Again, for typhoid and cholera, there is no good data at all. But in Bengal, where one of my colleagues is, that there's a lot of this LTOR, Vibrio cholera, they find by vaccination they can save a lot of lives. And the incidence of atopic disease hasn't increased. So I think these pathogenic, virulent bacteria and fungi, vaccination is good. And so it looks like those are okay. But I think antibiotics is different because it wipes out the good and the bad bacteria in the gut.
Thank you very much, sir, for an excellent session. And I would request all the delegates to please give a big round of applause. Thank you, sir. Thank you for your timing.
Thank you.